Okay, thank you very much. So, yeah, thank you, the ANERIS project and you for the invitation. It's very nice to be here. And actually, it was super cool to have Genev's talk before mine because he introduced many concepts that definitely I won't have time to introduce. So I'm a postdoc fellowship in Henrique's lab here in Portugal, and we work on analyzing microscopy data and developing tools that are connected to the microscope, as you can see in this image. I would also like to give the credit to the original authors, Luc Chamier et al. for the image. So as you could see, some flavors on the previous talk, AI-enhanced microscopy imaging is getting quite exciting. So it's true that these tools, they have proven an incredible potential, right? To segment the images with a very high accuracy, to denoise them, to find objects automatically from in very large scale data set, denoising and so on. But I would like to highlight the fact that this technology is bringing us a lot of power to go some steps further. So we can actually use the fact that networks allow us to enhance very specific signals from the images that we are analyzing, like different channels or information that we will double with fluorescence, for example, or with different colors depending on the optical setup that we are using to image. We can actually enhance resolution. And well, here you are seeing some mitochondria and some cells, but of course this happens even in your smartphones if you are using an iPhone to correct the quality of the pictures that you just took. So what I'm showing today is really oriented to microscopy, but it's generalizable to any sort of imaging setup. And one of the fields that is in which AI is becoming also very interesting, I won't have the time to speak about this further, but I will be very happy to discuss about it later, is data-driven microscopy. So how we can actually use all these segmentation or image processing tools to integrate them into the hardware and allow more, let's say, less biased imaging from larger field of use, or to decide when to actually we take an image or identify discrete events in our sample from a very, very large field of view. So this is very cool, but let's try to understand a little bit how this technology works so we can highlight the challenges. So the reason why deep learning has posed a new paradigm in image analysis is basically the way in which we are managing to get these tasks. So classically, what we would do is to define an equation or to define a function that has some parameters that we need to find, in which an input image will very, very implicitly give us a very specific output. But the problem with these classical algorithms is that we needed to know either the physics or the math behind to define this function. In machine learning, what we have is a completely different approach. We have the input image we want to analyze, and we have what we would like to have as an output, and it's what is called the ground truth that you see here. And what we have is a series of operations that are called networks, which is a series of many convolutions set in from a very high level perspective that we need to train. So by showing pairs of input and output, this model will learn what is the very specific processing we need to do to the image in order to denoise it in this case. And once we run the training series, what we say is that we have a trained model that we assess and we are ready to run predictions. And this is actually quite powerful because once the model knows how to process the data, we can automatically and very, very, very quickly analyze tons of images. And this is actually powerful. So the very exciting part in microscopy, and I would say in general in any image-driven research, is that we can play a lot with all these technologies. So I like to say that imaging in general starts from the beginning when we decide what is the imaging setup that we are going to use to observe. And the reason is because Here, we make some decisions on whether we are going to use infrared technology, we are going to use a normal camera, super resolution techniques, whether we want to get high throughput data or high content or multispectral. And this is the image acquisition setup that will then lead us to do the processing, right? And here is where we need to decide if we need to clean the images. And actually, it was very nice to have the introductory talk, because you saw how you need to pre-process all these images, right? To for example, correct color aberrations, correct the background, identify the small objects and the big ones in your image, so then you can analyze each of them independently. And all these steps, usually, or I think back in the time, we used to run them after we were acquiring the images. But my recommendation is that if you have some power on deciding how you acquire the data, or what are the techniques that you are going to use to get the image data, try to get some sort of loop of collecting some samples, process them, and then if you need, get back to the initial point to actually get data that it has quality enough to give you the information you need for your research. And of course, after this, we have the data analysis, right? So this could be an imaging kind of setup. And the exciting part is that all this processing is a deep learning playground. And I would like to highlight that we have many, many open source tools, not only for microscopy, that can be used for each of these steps that I'm showing you here. So this is very cool. But then the question is, okay, how do we make this deep learning technology accessible? And this is a very, very important part of my research and what I'm gonna try to speak about today. So from a very, I would say, personal perspective, I think any tool that we design to make this technology accessible needs to fill three very big gaps. The first one is that it needs to be open source, because these algorithms, they develop very, very fast. And if we wait until industry develop them in a safe way, we can delay a lot our research. Of course, the tools we develop, they need to be reliable for naive users, for people who are not expert in the algorithms, but they need them to pursue their research. And they need to be user-friendly, because otherwise there's no way to translate them into sciences, right? So this might be obvious, but bringing some history, it was not that much. So back in 2015, it was when the unit, this is the first very, it was a convolutional neural network, an encoder-decoder that made an inflection point in the way we analyze data. So back this year, we already had very powerful user-friendly tools for image analysis. Some of them, like Elastic or Weka, were already using machine learning, but the computer scientists developing these deep learning technologies, the way in which they were releasing their code was with this README file with very few instructions to actually be able to reproduce the code. And I was doing my research stay in the lab of Thomas Brooks, and it took us with the original developers of the code 2 weeks to actually build this code and be able to use it on my data. So this is not really working very well. In 2015, we have papers we called, this is a very important initiative in computer vision, just enlisting all the good technology with open source code. So this was already in 2015. In 2018, I said, okay, this was my paper. I developed a deep learning algorithm to segment some extracellular vesicles. And I said, okay, I want biologists to use my code, right? So I wrote very, very long instructions in a README file to show how to open a terminal and run my code. And I remember we had a collaboration with a biologist, and she told me, Esti, I'm not going to do this because this is already too complicated for me. I don't know where can I fail. I don't feel comfortable. So this was not working yet. And by this year, we already had some very cool tools that try to face this challenge of user friendliness, and these were CSV Deep, StarDist, DeepCell, and DeepLabCat. And then in the following years, we start having many different projects that try to actually bring this technology, fill in all these gaps. And nowadays we have a very, very, very nice ecosystem of machine learning user-friendly tools, and actually a lot of documentation and training courses from the community. So if you really want to use these very, very nice tools, I think almost anyone, not only from the microscopy imaging world, can start doing this. So now, because I know it's early in the morning, I want to ask you something, and I want you to think about it. Maybe you can write the answer in the chat. We will get back to this. So if you could ask for a future technological advance around image processing in 2 years from now, what could it be? And it doesn't need to be for microscopy, it can be for anything. So I would like you to think about this. And this is actually what a group of people thought about 2 years ago. And it's how a very nice project was born, and it's called AI for Life. So this is a European Horizon project composed of 10 partners from 8 different countries. And the idea is to bring artificial intelligence models and methods for life sciences. So what we want is to bridge this life sciences community with the computer sciences community by bringing infrastructure, by bringing user-friendly models and standards. So actually we can exploit the technology. And something that it's, I think, quite interesting from this project is that when we speak about community partners, we are not speaking about research groups, but about tools that are actually trying to face this gap between life sciences and computer sciences. So we have still 2 years to work on this, but I will try to introduce today the advances that we've been working on in the previous years. So what we wanted with AI for Life is to allow deploying, training, sharing deep learning models for image analysis. And for these, there are different challenges that we need to address, but the ones related to accessibility are related to the technical skills we need to deploy these tools, to the high computational resources we need to access on. So you saw the terabytes of data that many times we need to analyze, and this is not straightforward to do it. And of course, we need data, right? So I will try to show how we address these challenges. So if we think about what is a pre-trained model, is basically an architecture, as I told you at the beginning of the talk, of many different operations that we call convolutions, for example, and the parameters that we need to apply for each of these, let's say, filtering. The very cool thing is that once we train the model, this is a unit trained for segmentation, it's frozen, and we can encapsulate like in a box that we can actually just send it in a file and run it on new data. And this is very convenient because then we can very easily export it and use it everywhere. So in 2018, this was a very great idea from Arrate Muñoz Marrutia, my PhD supervisor, and Daniel Sachs, that will give a talk next week in this same workshop. They said, okay, this technology is very easily transferable, so why don't we try to bring it into Fiji? And Fiji, ImageJ, is a fully equipped scientific image visualization and quantification software. It's, if not the most famous one, is among the most famous ones for image analysis, and it's actually a very, very rich ecosystem. So together with Carlos García López de Haro, that is the main developer of Deep ImageJ, and Way, we developed a plugin for this Fiji software, in which you open the image you want to analyze, you select a model you want to run or you want to use to process the image, and it will process for you in a non-code fashion, regardless whether the model is in TensorFlow, in PyTorch, or whether you are in Linux, Windows, or Mac. So this was a very cool project, but then there's the question of how can I apply it to my data, right? I need new data to analyze. And then the answer is, well, you need to retrain these models and you need to fine tune. So here is where we start getting into a big bottleneck. So training most of these deep learning based image processing workflows requires some Python code. And the question is, how easy can it be or how difficult it can be? So I'm gonna put you here an example. So This is the GitHub repository of cell pose, and it's a very, very powerful tool to segment cells in microscopic images, and it's actually one of the best documented, I would say, software for cell segmentation. So if you go to the installation, not the training, but the installation instructions, you will start seeing many lines of steps that you need to follow or things that you need to consider. It will start speaking about GPU compatibility. Do we have a GPU or not? I mean, many times this is a question that it may look obvious, but is not obvious for someone that is not expert with these hardwares, right? Whether if you have Windows or Linux is okay, but if you have an M2 macOS, then you may run into problems. It starts speaking about CUDA, NVIDIA drivers, and you need to go to the NVIDIA website, try to download whatever you need. So actually the installation is already kind of complicated, and we still didn't arrive to the training, and there are no proper instructions about how to properly train these networks. So there should be a way to make this easier. And this is a challenge that Luc Bonchamier, Romain Le, Guillaume Jacquemin, and Ricardo Enriquez try to face with zero-cost deep learning for microscopy. So this project, they brought the full user-friendly experience, a user-friendly deep learning experience into the field. And the way it works is very, very simple. So you have Google Drive and Google Colab, right? So you upload your data to your Google Drive. You open a specific notebook for one of the tasks you want to train a model for. You train your model using Google Colab that allows you to use some GPUs for free for some hours. It runs the inference, you assess the model, you download, and you have your model ready to analyze the data. So this was very cool. They provide many notebooks for different tasks, for denoising, for classification, for detection, segmentation, super resolution, and all the notebooks, they have the code hidden. So actually you don't need to program, you just need to provide some parameters such where are you saving your data, where is your ground truth, what is the training and the test images, and all of them, they follow the same unified structure. So once you learn how to use one, you can use all of them. And this was really a very important step to bring in this technology. And now what we want is to move away from Google Colab and allow using this in local computers or workstations. And this is a new project that we just launched 2 weeks ago, Deep Learning for Microscopy Everywhere. Deep Learning for Microscopy made flexible, shareable, and reproducible. And what we want is to exploit something called containers that is similar to virtual machines that will allow us to basically, once you choose with a very easy to follow interface, the notebook you want to run, where are your images in your local computer. So you don't need to care anymore about privacy issues with Google Drive, for example, or you can use basically all the technology that you have in your research institute to access these terabytes of data that usually they need protocols to share the data and these sort of things. And then you choose where you want to save your results. And the software will automatically in the backend, right, check what is the workstation you are working with, whether it is Mac, whether it is Windows, Linux, whether you have a GPU or not. And it will run these virtual machines called, we are using Dockers, with all the dependencies for your notebook encapsulated there with the paths to your data, and will actually open a notebook that is very similar to the zero-cost notebooks, but now it's running on your computer, right? And this is all done safely. So of course, it has benefits for developers because, again, we are allowing them to disseminate their tools in a user-friendly manner and in a safe way by making sure that these containers and the dependencies are there and enabling this long-term reproducibility. Just so you know, in proprietary platforms, many of the deep learning packages are not always supported, or they may have updates that will basically break all dependencies. For users, we are still enabling them to use this technology in a safer way. And of course, we are contributing to this reproducibility and transferable kind of standards And something that I think is good, well, with these virtual machines, what we do is we pull them in a Docker hub, so anyone can download these virtual environments to keep developing technology. We provide models to the zoo that I will speak about it very, very soon. And then, because we wanted to expand from zero cost, what we develop is a way, if you have your notebook and you have your model, you can actually use our platform to read what are the dependencies that you need for your code, and it will create all these technological pipeline to encapsulate your work and provide it in a user-friendly manner to the user. So this is very nice. And now the last part of these challenges that I told you we wanted to address is how we share all this technology, right? So this is cool, but how do we make it available for the community? And this is what I call the fairy deep learning ecosystem, the findable, accessible, interoperable, and reproducible fairy deep learning. And in particular, it's an initiative called the BioImage Model Zoo that is led by Wei Ojian, Florian Jack, and Anna Gresham. So what is this about? So the BioImage Model Zoo is a website where we have pre-trained models, and these are displayed by cards. And these cards, they are you can interact with them. So you can download the model, you can see the licensing, the DOI, the publication that the model is related to, the notebook that was used to train a model, the training data. So you have a fully documented model or deep learning model tailored for life sciences. And it also allows to directly interact with the developer. So you can use GitHub issues that are linked to the model to say, hey, I'm not able to train this model, or it's not working for me, or what should I do? So this is very cool, but this is still not fair. So one of the very big pillars is the interoperability. And that's why it's a key feature of the BioImage Model Zoo, and is where I want to highlight that this is a work we are doing from the community for the community. So what it means is that the BioImage Model Zoo is formed by all these open source software for image analysis, and the people who is working behind this is the people, the researchers involved in all these softwares. And what we want is a format that is able to run on all these software in the same way. So basically, the Stardist model that Jenny, for example, was showing to you, 2 segment cells, you can run it in Fiji, you can run it in QPath, you can use it in the notebooks, you can use it in Stardist and so on. So we want models that are interoperable so you can benefit from the features of all these software and choose the one you want and not be limited by the model itself. And this was a very, very kind of difficult race, because what we need is then to define a standard, right, for this model. So we need to very technically describe the model itself. So how should be the input image, the input shape, these mathematical dimensions of the input? How do we normalize the data? What's the receptive field of the network, the post-processing, the output shape? So all these technical geekiness of the model. So we can actually, when we give a new image, it will process it, even doing this puzzle that we call tiling, right? When the images are very big, we need to process them by different batches. And of course, because this is for life sciences, what we want is to have models that contain example images, but also the metadata of the image. So what is the pixel size? What is the shape? What is the modality? What is actually the living matter that we are seeing in this image? And this is possible by something that we call the resource description file. So it's this text file, a YAML file, that when we have all this information kind of in a text format, we can design programmatic tools to read it and be able to deploy these models everywhere. So what we provide now is Python and Java utility. So one is the BioImage IO core library, and the other one where Janif did an impressive contribution is called the Java Deep Learning library. And with these 2 programmatic tools, what we allow is to consume this format in all this software and from any of the deep learning libraries that we use to train the models or to create these models. So of course, once we have tools, programmatic tools to access or to these standard interfaces, right, to consume the models, we can enhance their reproducibility. So we can start enabling model fine-tuning, new model contributions. We can design user interfaces to manage the models, similar to what we did in deep imaging. And now you will ask, well, and why is this useful, right? So this is a huge amount of work, but how can I benefit from this? So I'm going to show you some use cases. So I told you that we need data, right, to analyze the images. So something that is very time consuming is to actually label the data, annotate it. But once you have pre-trained models, you can, for example, choose the one that was trained to process images that have similar features as yours. So let's say, for example, here, we want to segment the edges. So we can pre-process the images with these pre-trained models, then curate the results and create new training data, but much, much faster. Then we can fine-tune this model with the new data and use it again in the same software as Elastic or in a new one and run quantification. But we can go one step further, and is when we start providing tools to scale very well to the huge amount of data that we are recording with our optical setup. So here I'm showing you a 3-volume embryo. It's a beetle embryo. And what we usually would like to do is to segment each of these nuclei and then track it. And you can imagine that manually annotating these gigabytes of data is not really human. There's not enough human power to do that, right? So what they propose in the cell tracking challenge is, okay, I'm gonna just give you some partial annotations of some 2D slices, and you should be able to train an algorithm that identifies them, because basically there's a lot of redundancy in the data. So because we know that we have pre-trained models like StarDist, we can actually use the fact that these models already know more or less how a nuclei looks like. We fine-tune with these partial annotations, we segment each of the slices, and then if we run this model trained already in Fiji, we can use connected components or TrackMate to reconstruct the 3D volume. And very easily, without coding, we can actually go from here to here in few steps in just one day. So this is very cool. Then another thing we can start thinking of is about domain adaptation. This is a bit more technical, but it's something in which Anna Kresh's lab is doing quite a good or interesting research. So here, what they propose is, okay, let's say you want to segment mitochondria in these electron microscopy images. So you can try random forest. So you can manually label what is a mitochondria, what not. And this random forest will give you a probability map. So the distribution of this data is already normalized and is in the space of probability. It doesn't matter anymore if it is electron microscopy or if it is fluorescence. So then we can actually use a model that was trained on this data distribution, on these probability maps, to actually enhance and identify where are the objects of interest to segment. So with this, deep learning is quite cool, but I really like this slide from Joanna Pilvainen, where she says it's not a quick fix for image processing. So actually it's full of challenges, and I would really like to highlight some of them and some of the things we need to be aware of when we do this technology. So the first of all is that we need to identify the objective that we want to train the model for, right? So what is the question? Do we want to segment? Do we want to identify the object? What is really what we need to proceed our to do our research? And usually what we would do is, okay, is there a model for it? If there is, we can use the bioimage model, for example. We need to assess the network, and if it is accurate enough with our data, we just run the inference. If there's no, we need to customize the network, right? We need to train it. We need to build the ground truth. We need to evaluate again. And if it is accurate enough, good. But many times it might not be. So then is it because we need more training data? Is it because we are actually evaluating properly the model, or is it because the objective was not correctly defined? And these are the very, very important 3 points that we need to point to. And I think the community is doing a great effort on providing guidelines to run your bioimage analysis, providing awareness of the way we need to evaluate the data, and the fact that in this world, there's no one solution that fits all. So this is actually quite nice. And I would recommend reading these works if you want to start playing with it. So then there's I said the objective is one thing, then the image data quality. So the thing is that we don't always have annotated data, right? So there are more and more research, there's more and more research going on in this sense. So we have the typical supervised learning where we show what's the ground truth and then we train the network with that. But we start having unsupervised kind of approaches, self-supervised. So depending on the nature of your data, if you are able to label the data or not, or you can only very weakly identify the points you want to segment, you will need to run one strategy or the other one. And here I'm showing it for microscopy, but it happens actually everywhere. The approaches we use to obtain the data will already limit the quality of the data, right? It will limit the resolution, the temporal resolution, the contrast, the signal itself, the signal to noise ratio. And what we need to find is usable data, not good data or appealing data, and usable data for the algorithms we are going to run on this. And here is a perspective that I will also recommend reading very recently. Joanna and Guillaume, they publish it and they guide through all these steps. So here I'm showing you just a very easy example. So if we want to segment these nuclei, we have many different resolutions at which we can acquire the data, but all of them will be at the cost of huge data sets. We are speaking about gigabytes, or a smaller or larger field of view. But actually, we segment these nuclei, the area, it will be the same no matter the resolution. However, if we try to segment these mitochondria, we will need to very carefully decide the resolution, because not all of the qualities will allow us to identify different structures. So in this sense, the last thing we need to take care of, and here are some works that I will recommend, is how we validate the model. So here, imagine that you want to segment these mitochondria, and if you want to get what's the average fluorescence intensity, you can get a circle around it, right? And this identification will be enough. But if you want to identify the length, curvature, diameter, this segmentation as a circle will not be valid. You will need to actually segment the orange part. So there are no good or bad kind of models. It's more whether your approach is valid or not for your research. So now, very briefly, because I'm run out of time, I would like to highlight what's the vibe in the community. So first of all, I think most of the people we are working in, we are really, really involved in training. And what I think we are seeking to empower the life sciences community with this technology, to actually bring all this image processing technology into the lab. Then, of course, there's not only awareness of these, all the things I told you in the life science community, but also the computer sciences community. And here, I want to speak about the experience with the cell tracking challenge. So it's been running for 10 years. Basically, they have many data sets that they propose to develop methods and benchmark these methods on their accuracy. And a few years ago, we said, okay, many of these tools are not usable by lab scientists. So let's propose some reusability guidelines. So let's write some guidelines for the developers to tell how to make their code more easily replicable or reusable with new data. They were optional, but all the computer scientists followed them, all the participants. And this was it was quite exciting, right? Because you realize that there's also awareness of these needs in the other side of the bridge, right? And finally, I think we need to start working more together. We already need it, but now even more, because these AI methods, they start embedding not just the scientific knowledge in the processing. And I think this year we got a very impressive development called Segment Anything. So probably you are already aware of this model, where you can very, very briefly or very sparsely tell, okay, I want to segment this thing here just with a point, and it was segmented for you. And Konstantin Pap, he's a PI in Gothenburg University, he already managed to embed this into Napari and make it actually usable for microscopy. So with this is all. I will